Okay, let's start part 2 of semester 2. We're going to pick up right where it says here, protein synthesis. Remember, protein synthesis is all going to be about taking the DNA out to the protein, right? Or in essence, we're talking about taking a genotype and turning it into a phenotype. Now, in a eukaryotic cell, which is what I'm going to mostly focus on, we have organelles that are in place, and those organelles cause some differences in the transcription translation process. The eukaryotes have a nucleus. We also have some editing that we'll talk about as well. Prokaryotes will not have those pieces. So DNA to protein, that's protein synthesis. Let's start with the first piece, which is transcription. Don't get transcription confused with translation. Trans with a C. C becomes before L, so transcription comes first. This is going to all be about going DNA to specifically mRNA. This is going to happen in the nucleus of the eukaryotic cell. And we have a couple different things that play a part. One is RNA polymerase, which by its name is going to be making the mRNA strand. So it's going to be arranging those mRNA nucleotides in the proper order. It gets that information from the DNA, the gene that it's trying to transcribe. The Tata box and the promoter region is the location where RNA polymerase is going to attach. We'll connect this to an Operon here in a second, but that is that initial attachment location. So this is going to be upstream from the gene that we're working on. Just a little below that gene, there might be an operator where a repressor will attach to, and that can actually govern whether or not the RNA polymerase can move downstream to get the gene. We'll talk about that in the next slide. So RNA polymerase moves 5' to 3', just like DNA polymerase, but it does not require a primer to attach to. It's just making mRNA. It's going to shoot down. It's single-stranded, so we don't have to worry about a leading, lagging strand. It's just going to be leading. And that piece that comes out, that mRNA is, for a eukaryotic cell, going to be considered pre-mRNA, where you have to do a little bit of editing to make it functional for the ribosome to actually transcribe it or translate it. So operons, this is where we're going to tag in our operon discussion, just a real quick overview. So one is your RNA polymerase. So just like we were talking about in the last spot, RNA polymerase is going to make that mRNA for us. So this area down here is going to be the gene of the interest so that we can make the protein that we're looking for. Number 2 is the repressor. Repressor, holy moly. And that is attached to the operator. And this 3, that's that promoter region we just talked about. So that would be where the Tata box is. That's also going to be maybe cyclical AMP is going to help RNA polymerase join into that location in order for it to move downstream. But that is the guiding sequence here. And then you have these little pluses or these cross pieces, and those are the inducers. So in this picture, that inducer is pulling off the repressor so RNA polymerase can move downstream. If you remember, we talked about this from the lac operon perspective, where if 6, 7, and 8 were the gene to make lactase, where we want to work on lactose, lactose might be the thing that pulls the repressor off, because we wouldn't want to break lactose down unless it was in the environment, right? So we wouldn't want to make the enzyme to break the lactose unless lactose is present, right? Otherwise, it's energetically taxing, and that's going to decrease your fitness and evolution, right? So that is a big part for our operon discussion. We can discuss that more if you have questions about those things. But then, after we make our mRNA, we also have to process it. So remember, this is done by a spliceosome, which is a combo of snRNA and snRNP. So those two things work together to make this big, huge cutting body, which will find the beginning of an intron and the end of an intron so that we can cut out the intron and actually glue the exons together. So your result is your mRNA after this is going to be shorter, and it will just have exons, only exons. So the problem is prokaryotes, they don't do this. So if we are going to make a prokaryote do something for us, and we want to put our gene into a bacteria for them to maybe synthetically produce insulin for us, then we actually have to get that RNA to be processed first and then put it back into a DNA form. And that's where we use that enzyme from HIV, where it does that reverse transcription process, and that's why it's called reverse transcriptase. Okay, so we would use that process if we wanted to help out a bacteria. And we'll talk a little bit about that as we move into DNA tech. And then we had the hippie video, right, where we got the people rolling around the football field, giving you the translation. So this is from the mRNA to a polypeptide. Right, and that takes a couple steps, but it's going to involve a ribosome. That's going to be the active place for that to happen. Remember, ribosomes are made out of rRNA. We have the mRNA, which is bringing the code, and now it's been edited. We also have tRNAs, and the tRNAs are going to be bringing the amino acids. Now, remember, the mRNA has a 3-section sequence that everything's going to be read in, for example, like AUG, and we call those codons. The tRNA will have a 3-section complement that goes with it, and that's called the anticodon. So whatever the tRNA or the complement will be, remember that this is RNA, so there will be no T. So A would go with T, but instead it goes with U. And so you see the anticodon for the tRNA would be that UAC that goes with the AUG. So we keep reading down the sequence. It's called an indexing process, and in turn, we keep getting more and more amino acids coming in. Let's kind of look at that. So this is what it kind of looks like in a stepwise fashion. So the starting picture way up top here, if you remember, we had like initiation, which that mRNA comes in, the bottom subunit attaches, then we get the first tRNA binds in and only binds in at the P site for that first one, and it brings the amino acid that goes with AUG, which is MET, methionine, and then the second one that comes in is going to come in at the A site. So then the second, we get the top subunit that drops in. That's initiation. Then we start elongation. So then the second tRNA comes into the A site. We get a bonding situation, and then we index, right? So you can see that we get this bond over to this second tRNA, and it's holding now the polypeptide chain. It indexes, shifts. The empty tRNA exits at the E site, and the polypeptide is growing at the P site. So A attach, P polypeptide or protein, and E exit. So you keep doing this and keep doing this and keep doing this until we eventually hit the stop codon. The stop codon is going to cause the release factor to join in. The release factor does not have an amino acid. So remember, there's that big, huge chart, right, that had all the different amino acid coding pieces, right? We had all the amino acids. That's fairly universal. So if it's AUG in an animal, it's going to be AUG in a plant. It's going to be AUG in a fungus. But that code will allow us to understand what amino acids go for what specific genes, help us understand how actual organisms develop the way that they do, right? And that's kind of where a lot of the hot biology is right now, is figuring out that developmental piece. So then we moved into, well, what about proteins that stay for that cell versus one that use the endomembrane system? You remember the endomembrane system we talked about in the cells unit. That's where we talked about how a protein either stays for that specific cell or it's exported for a multicellular organism to be used in other places. For example, the insulin that's produced by your pancreas is used all over your body, but it's only produced down there in the islets of Langerhans of your pancreas. So those cells have to produce the insulin protein for the entire body. So it's done through this endomembrane system. And what happens is at the end of that insulin, there's a marker piece that this SRP, which is a signal recognition particle, will attach to and allow for it to facilitate into the endoplasmic reticulum. So we've got ribosomes that are in the vicinity. It looks like they're always attached to the ER, but they're kind of floating in and out. Whenever we take a picture, that picture is just for that given time. So it's going to look like the rough ER has ribosomes attached to it all the time, but it's not actually the case. They're floating in and out, and these signal recognition particles are going to be the pieces that help facilitate that polypeptide chain actually into the ER, which then can keep getting folded as it moves to the ER, buds off through a vesicle, goes to the Golgi apparatus. Golgi apparatus modifies it, tags it up, bounds out again through another vesicle, binds to the cell membrane, and then exports out of the cell, maybe into the bloodstream or the vicinity, so that other cells can use the product from that one specific cell. So that is a really important piece at the end of protein synthesis, if in fact we are going to make something for other cells. Now, prokaryotes, they don't have to necessarily do that because they're one cell asexually reproducing. They're not doing that situation unless, of course, they're communicating, right? We talked about sensum quorum and how cells can talk, bacteria can talk to not only their own species, but other species that are bacteria. They can mount an attack. If you remember Dr. Bonnie Bassler talking about that, it was fantastic, right? If you don't remember that, look up that TED Talk again with Vibrio fischeri and their bobtail squid and how they do all that stuff. And then we started talking about plasmids, and plasmids being circular pieces of DNA from bacteria. And we talked about how we can engineer them. So we can actually put in genes we want into those plasmids. So in our lab, we used restriction enzymes to recognize the ends of a gene that we want to cut. So here, the example is EcoR1, which stands for E. coli restriction enzyme number 1. Notice in the picture, it's all EcoR1. You could use other ones, like there's one that's called BAM3 and HIND3, and they've got all kinds of Roman numerals, and they got all kinds of different bacteria that they come from. Each one is really specific. So you can see in this picture, it's recognizing the sequence GAATTC. It's a palindrome because it's the same forward as it is back on the bottom side. But it's going to find that exact sequence, and it's going to cut between the G and the A. And in this case, it makes it jagged. And that jaggedness allows for what we call a sticky end. And sticky ends is just a generic term that means that there's open areas for hydrogen bonding between another sticky end that would have that complement. And so if you cut your plasmid with EcoR1, that ring opens up, so it's no longer a circle, it's now an open ring, right, like this. And then if you cut out a gene that you know has the same restriction enzyme locations on either side of that gene, you could cut it out. So in our lab, we use the jellyfish glow gene, that GFP gene. We cut that out, and then we stuck it into the plasmid, right? Now, not all of them worked, and some of them didn't, so that's part of our screening process. So in turn, when we stick those two pieces of DNA together, it's called a recombinant piece of DNA, because we've recombined two different pieces of DNA together. Now, if we allow a bacteria to suck that up from the environment in a naked fashion, we call that transformation. And that's where our lab kind of took off, because we then screened for it, and we screened for it with an antibiotic. Because remember, antibiotics kill bacteria, not viruses. And that antibiotic resistance can be found in the plasmid in a different location. So if the bacteria actually took in the plasmid that we put in its presence, it should survive that antibiotic environment. And if it didn't, then it didn't take it up. And that's where we got those 4 different scenarios where we had the LB, the LB amp, the LB amp with the plasmid without it, and then the LB amp and ERA. And the ERA part, the rhabinose, was connecting back more to the operons. That was the inducer that got that GFP gene transcribed and translated so we could actually see its phenotypic effect. Okay. One more slide, and then we'll kind of finish up with this video. And we'll pick up more DNA technology in the third video. Using restriction enzyme, you can cut DNA. So this is kind of the crime scene stuff that we would like to identify, or if you're trying to figure out the differences between different animals or plants, you can use this technology. Discovering diseases. There's a lot of different purposes for doing what is called electrophoresis in a southern blotting situation. So say you have an individual that has 2 different pieces of DNA, right? So not identical twins, but two individuals that have different DNA. We're just going to use this picture down here for the genotypes to just kind of show you. So if you're a person that is big A, you have two homologous pair pieces of DNA that should look exactly the same because they're the same genotype. If you have an individual that is little a, they would have two pieces of DNA that would be different but the exact same. And then you have a person that's big A, they'll have two pieces of DNA, but both pieces are going to be different from each other. And so if we add a restriction enzyme, remember restriction enzymes are incredibly specific. So they're going to go after a very specific sequence. They would cut, let's say that big A, they cut it and cause a big piece to show up, or they don't cut it at all right? So maybe it doesn't cut it at all. And in turn, you get a band that indicates that large piece. The little a gets cut different, and maybe you get some smaller pieces out of it. Sorry, this was the big A, little a one. Sorry. And this one would get cut in two different places. So you get two different bands for the big A, little a. We could do this for all kinds of individuals. You have a crime scene, and you have two suspects. The crime scene, you find this banding pattern because of the restriction enzyme for the sample that you have. Individual 1 gets cut with the same exact restriction enzymes. They get that banding pattern, and then individual 2 ends up matching the unknown. This is a way that we can indicate whether or not a sample came from the unknown source. So this is what we do for crime scenes. We do this for diseases. The only added extra pieces, just because those bands are in the same location. So if we look at this picture up here with the gel, all these bands are at the same location. That does not mean that they're the same sequence. That just means that they're the same size. Because remember, gel electrophoresis separates DNA based on size. So the smaller pieces move to the positive end much more fast than those that are bigger because they're being pulled through this agarose matrix. So they're going to be moving through a much swifter rate than those that are big. So the big ones up top, right? So these big ones, just because they're all the same size does not mean they're the same piece of DNA. So what we can do then is search for a sequence, and that's where that southern blotting technique comes in. You make a radioactive probe of a sequence that you know exactly what you're looking for. You wash it across the gel, and then you pull the DNA into a film that you can actually run through an X-ray device to pick it up. And so we can see kind of in this picture here, we have the gel that made this great band pattern, but then we look for where the sequence is, and you can kind of see in this picture down here, it shows up in 3 different individuals in 3 different spots. So it might be a duplication situation, might be an error, might be a mutation, but we're looking for specific sequences with that southern blot situation. And that's video number 2. I'll pick up with video number 3 with a little bit more DNA technology. We're going to move into some evolution after that.